Okay, so welcome back, and without further ado, let's go ahead and cover what we're going to be going over today. So, today we're going to dedicate time to talking about the support structures of the central nervous system, and then we're going to draw a focal point to the anatomical layout and the functional characteristics of the spinal cord. Now, as always, there's an associated lecture handout. I'd pull it up either as a PDF and follow along, or I'd have it printed out and be writing along as we go along. Pause where you need to. That's kind of the nice thing about videos. So, our key points today is we're going to start by talking about the meninges. We're then going to shift our focus to the ventricles and the cerebrospinal fluid flow pathways, or CSF pathways. Then we're going to introduce some terminology that's going to come up over and over again throughout the course of not only this class, but anatomy and physiology classes later on down the road. Then we're going to talk about the spinal cord, and we're going to draw our focus to the white and gray matter structures of the spinal cord. So there are your learning objectives, and when we think about the meninges are connective tissue structures that stabilize the brain and spinal cord. They are support structures of the brain and spinal cord, and they are connective tissue structures that wrap around both the brain and the spinal cord in one cohesive unit. And what they do is they support not only the brain and spinal cord, but things like the blood vessels and nerves that feed into the brain and spinal cord. They also provide a cavity through which critical fluids like CSF can bathe the brain and spinal cord. So when you think about the spinal cord, for example, we know the brain and spinal cord make up the central nervous system. The meninges surround and support the brain and spinal cord, so we call them stabilizing and protecting structures of the central nervous system. When you think about the spinal cord, an average spinal cord is about the thickness of your pinky finger. And within a structure that's about the thickness of your pinky finger, you have 100 million neurons carrying information to and away from the brain. There's more information that travels along the spinal cord in the course of a day than there is digital communications that happen within the United States over that same period of time. There's thousands and thousands of pieces of information just going up and down the spinal cord every second. So the spinal cord needs a lot of support. It's protected by the vertebrae and the spinal cavity that the vertebrae form, or the vertebral cavity, whichever one you want to call it. And they're also protected and supported by a layer of connective tissue referred to as the meninges. Now, I'm not going to talk about the different layers of the meninges using this image, but here we have the spinal cord, and you can see the spinal nerves branching off. Remember, it's about the thickness of a pinky finger. And then you have these connective tissue structures surrounding the spinal cord. Collectively, we refer to those as being the meninges. So on an exam, if I said identify the general set of structures, the connective tissue that surrounds the spinal cord is referred to as the spinal meninges. The connective tissue that surrounds the brain, and it's one continuous set of connective tissue that the spinal meninges essentially turns into the cranial meninges. The connective tissue that surrounds the brain when it's in the cranial cavity is no longer called the spinal meninges, it's called the cranial meninges. Now, again, the meninges are extraordinarily important for not only stabilizing the position of the brain and spinal cord and the blood vessels and nerves in that particular region, but in providing a cavity for fluid to flow and bathe the brain and spinal cord. So when you look at an image of the spinal cord, we've got a cross section here, we have a vertebrae. You can probably figure out this is posterior by looking at the spinous process, which is always going to project posteriorly, which are those little bumps you see in your back. And then the body of the vertebrae is always going to project anteriorly. Now, when we think about the layers of the meninges, the spinal meninges, before we even get to the meninges, because the spinal cord is so little and delicate, little tiny bit of damage to the spinal cord can produce massive consequences. We want to protect that spinal cord even more. And the way that body does is it wraps it in a layer of fat. Now, the area or the volumetric space in which this fat exists is called the epidural space. The fat in this space is called the epidural fat. So what you should be doing right now is on your image of the spinal cord in that cross section, you should be identifying the support structures. Now, the epidural fat in the epidural space is intended to cushion the spinal cord. It also stabilizes blood vessel networks in that particular region. Now, deep to the epidural space in the epidural fat, closer to the spinal cord itself, we start our meningeal layers, and the meninges are broken into layers and cavities. The outermost layer of the meninges is called the dura mater, not dura mater, but dura mater. It means tough mother in Latin. It's a layer of dense connective tissue that stabilizes the spinal cord in its position and stabilizes the position of spinal nerves as they leave or exit the spinal or the vertebral column through the intervertebral foramen. Now, the dura mater is the outermost layer, stabilizes the position of the brain and spinal cord and the associated blood vessels in that particular region. Deep to the dura mater is another layer referred to as the arachnoid mater. Arachnoid because the protein fibers, right, that project off of it form this kind of spider web appearance, and mater meaning mother, it means spider mother. So our next layer is the arachnoid mater. Now, deep to the arachnoid mater is the space that's most relevant to this particular class. There's a volumetric space that surrounds the spinal cord between the arachnoid mater and the deepest layer of the pia mater. That volumetric space is called the subarachnoid space. And the functional role of the subarachnoid space is to allow the flow of cerebrospinal fluid around the brain and spinal cord. And cerebrospinal fluid is really important because it's providing things like nutrients and oxygen to neurons. It's also taking away things like metabolic waste and carbon dioxide. So we need cerebrospinal fluid circulating around those areas, right? Because it's giving the neurons good things and it's taking away bad things. I've heard textbooks call it the blood of the brain. It's not really blood. It's a filtered product of blood, but it's not really blood. So if I was to say identify the volumetric space indicated by the pointer, that would be the subarachnoid space. If I was to say what flows or what substance would you find here, that would be cerebrospinal fluid. Now, the delicate network of connective tissue that actually contacts the brain and spinal cord directly is referred to as the pia mater. Now, the pia mater stabilizes the delicate blood vessel networks that feed into the brain and the spinal cord. It's a very thin, delicate layer of tissue that almost looks like wax when we do the brain dissections. And there we go. So we have the dura mater, the arachnoid mater, the subarachnoid space, and the pia mater. Within the subarachnoid space, we have the flow of cerebrospinal fluid. So this is the image that you have on your particular diagram. First off, where would the spinal segment be? I think that's just an interesting question to ask. Second off, we have our posterior and anterior regions. We have the epidural space filled with epidural fat, the dura mater, the arachnoid mater with that kind of spider-webbed appearance, and then contacting the actual spinal cord itself is the pia mater. In the space between the arachnoid mater and the pia mater, so this space right here, that volumetric space is called the subarachnoid space. What that volumetric space does, what that subarachnoid space does, is allow for the circulation of cerebrospinal fluid. Now, the spinal cord is surrounded by the spinal meninges. The brain is surrounded by what are called the cranial meninges. Now, when you look at the cranial meninges, you can kind of go through on your image and label these as we go through and think about having that out and available to you, for example, during a quiz. So when you look at the cranial meninges, they're also broken into the dura mater. For the intro class, don't worry about the paraosteal and meningeal dura. Just worry about the dura mater as being the outermost layer, the arachnoid mater, and the pia mater. So when we look down here, what we've done is we've taken a frontal section of the cranium and the brain, and we're looking at the connective tissue structures that are surrounding it. So we've pulled this out and we're kind of looking at this. Now, you have the dura mater on the outside, the arachnoid mater. The subarachnoid space is this volumetric area between the arachnoid and the pia mater through which cerebrospinal fluid circulates. That subarachnoid space is really important. Then you have the pia mater. That pia mater actually contacts the brain directly. It's that delicate thin little layer of connective tissue right there, stabilizing things like blood vessels that feed into our cerebral circulation. And that's all you really have to know about that for this class. Now, a couple of other ancillary structures I want to highlight to you while we're here is if you notice the way that these meninges fold on one another, they form a series of what are called sinuses. The sinus that runs sagittally and superiorly through the cranial cavity is what's called the superior sagittal sinus. When we're talking about the cardiovascular system, you can think of a sinus as being like a big fat daddy vein. And what this sinus does is it not only drains blood from the brain, but it also functions as a common drainage point where you get the transition between cerebrospinal fluid and blood. And the reabsorption of cerebrospinal fluid from the subarachnoid space is actually facilitated by these little structures right here called arachnoid villi. Some textbooks call them arachnoid granulations, but I'm going to call them arachnoid villi. ARACH Noid Villi or Villus if you're talking about singular. So what this is calling an arachnoid granulation, some textbooks call it that. I prefer arachnoid villi just because I like thinking of them like little finger-like projections. So another thing you get with the meninges is that they form a series of folds. So one of the, and pretty much the only meningeal fold that you have to know for an introductory anatomy and physiology class is the falx cerebri. So the falx cerebri is the fold that separates the left and the right hemisphere of the brain. What you're looking at here is the falx cerebri. Now, what separates the higher order brain structures from the cerebellum is another meningeal fold called the tentorium cerebelli. And if you look at these, you have the falx cerebri, this fold right here with the superior sagittal sinus running through it. And separating these higher order brain structures from the cerebellum is another meningeal fold called the tentorium cerebelli. You also have the falx cerebelli, which actually separates the two hemispheres of the cerebrum, or is a fold that kind of projects into the two hemispheres of the cerebrum, but we haven't really talked about that. So when you think about the meninges, they're really important from a clinical perspective. One of the reasons they're important is that they stabilize the position and have things like blood vessels running through them. If you clip a blood vessel and you start to get a bleed, that bleed can actually start filling up these little gaps or these spaces that you have within the meningeal layers. And in this case, the bleed is happening within the subarachnoid space. So if you have a bleed taking place and that blood is pooling up in the subarachnoid space, what it's going to do is it's going to put pressure on the brain. And it results in what's called a subarachnoid hemorrhage. So the subarachnoid hemorrhage has put so much pressure on the brain that it shut down kind of critical brain functions. And this person unfortunately did not make it, if you couldn't guess from the diagram that we're looking at. So you may have heard on like a crime show or something like that, cause of death is a subarachnoid hematoma. Well, that's what you're looking at right here. So we care about that. And there are symptoms that predate that. Like if a kid is playing football and gets hit on the head and starts presenting with dizziness and nausea and things like that, you pay attention and you make sure that there's not bleeds in that area. Another condition that we have to be concerned with that impacts the meninges and ultimately can put pressure on things like the brain and spinal cord is what's called meningitis. Now, meningitis is an inflammation of the meninges, which are the connective tissue structures surrounding the brain and spinal cord. There are a lot of different causative factors of meningitis. The most dangerous is probably bacterial. And there are vaccines for this. In fact, many universities require you to get a vaccine for meningitis, but it can also be caused by fungal infections, viral, parasitic infections. You have autoimmune meningitis. There's multiple causes of meningitis. And if you ever have a stiff neck, fever, and you're vomiting, just go to the ER. Just go, right? Because you probably, or you could potentially have meningitis, and that's just not something to play around with. Whether you believe in antibiotics or not, please, just under those circumstances, go to the doctor. Now, what meningitis does is the inflammation of the meninges interferes with the flow of cerebrospinal fluid, and it also puts pressure on critical brain centers. And we can talk about how that can produce, what's called autonomic dysregulation, where blood vessels aren't working properly and things like that. But suffice it to say, meningitis is a condition of inflammation of the meninges that can put pressure on your brain and your spinal cord. Now, when you look at this MRI image of meningitis, you can actually see that kind of compression of the brain and spinal cord in the actual meningeal space, and putting pressure on the brain is not a good thing. One of the ways we look for example, spinal meningitis is there are simple tests you can do. So a pediatrician may try and elicit what's called the Brudzinski's reflex or the Brudinsky sign, whereas if you pull up on the head, you get a reflex movement of the leg or a jerk of the thigh up. And it's not very sensitive, but it's very specific. But suffice it to say, meningitis is an inflammation of the meninges. Now, when we think about cerebrospinal fluid flowing through those meningeal spaces, that cerebrospinal fluid had to come from somewhere. And where that cerebrospinal fluid originates are in blood vessel networks that line these hollow cavities within the brain and spinal cord, and specifically the brain here, that we refer to as being ventricles. So the ventricles that are on your list to know, these hollow cavities through which cerebrospinal fluid flows, are the lateral ventricles, which feed higher order brain centers like the cerebrum. They're huge, they loop around, they have these horns. We're going to look at it from a different image. From the lateral ventricles, cerebrospinal fluid circulates through the interventricular foramen to the third ventricle. From the third ventricle through the cerebral aqueduct to the fourth ventricle. From the fourth ventricle, they're either going to squeeze out these little holes into the subarachnoid space and circulate around the brain directly. Those little holes are called apertures. Or that cerebrospinal fluid is going to enter into the central canal, where it'll pop out of the tip of the spinal cord at the conus medullaris and then circulate up, right, in that subarachnoid space. So when we think about the cerebrospinal fluid flow pathways, when you look at a sagittal section of the brain, what you see here are the lateral ventricles. So they're directly inferior and they come into contact with regions of the cerebrum, which form the outer walls of them. And these lateral ventricles are huge. That cerebrospinal fluid then flows to the third ventricles. The lateral walls of the third ventricle are essentially the thalamus and the hypothalamus. That third ventricular space is about right here through the cerebral aqueduct to the fourth ventricle, which essentially supplies or provides a circulation route for structures like the cerebellum and the brainstem. So you see the hierarchy of the organization of the brain through the ventricles. When we look at this image here, it's kind of nice because we can see the intact pia mater on top of the brain, that gentle little layer. But we have these microscopic blood vessel networks that line the roofs of the ventricles called the choroid plexus. They produce cerebrospinal fluid. That cerebrospinal fluid enters into the lateral ventricle through the interventricular foramen to the third ventricle, and then we don't see the brainstem structures here, the cerebellum, but you can kind of see what I'm talking about. So when you get that frontal section of the brain and you're wondering what these things are, you're looking at the ventricles, you're looking at ventricular spaces. Now, the classroom model of the ventricles looks kind of like an alien head. It looks kind of like this thing right here. If I was to ask you to trace the flow of cerebrospinal fluid, well, cerebrospinal fluid is ultimately synthesized by what are called by capillary networks. You push blood plasma across a capillary. Those capillary networks that line the ventricles are called the choroid plexus. That cerebrospinal fluid is then circulated, or filtered through ependymal cells. And once it exits those ependymal cells, that what was once blood plasma in the choroid plexus, once it enters into the lateral ventricle, it's no longer called blood plasma, it's called cerebrospinal fluid. So cerebrospinal fluid derives from blood plasma, it comes from blood plasma. You can think of it as being filtered blood plasma. So think of blood plasma as being sludge, right? Or kind of a mixture, a heterogeneous mixture of stuff, like if you were to put a bunch of dirt into water and mix it up. And then think of cerebrospinal fluid as that coming from that kind of water, that dirty water, but being filtered. And we filter that cerebrospinal fluid because we don't want the solutes, that stuff in the blood, getting into the regions that come into contact with our brain willy-nilly. So we have the lateral ventricles. That cerebrospinal fluid flows through the interventricular foramen to the third ventricles. Notice that the lateral ventricles are huge. They form these horns. So through the interventricular foramen to the third ventricle, which essentially circulates around the diencephalon, through the cerebral aqueduct to the fourth ventricle, through the central canal of the spinal cord all the way to the tip where it's going to enter into the subarachnoid space and circulate around the outside of the brain and the spinal cord. So we have the internal circulation of cerebrospinal fluid, and then we have the external circulation of cerebrospinal fluid until it's eventually drained. Now, think about this particular image because the lab model for this particular image is going to become relevant on your lecture exam and on your lab practical, which are kind of hybridized now. Now, when you think about the flow of cerebrospinal fluid in the central nervous system, let's see what we mean by looking at a sagittal section. And I've given you an image on your handout to kind of follow along. So if I give you a question that essentially said, which of the following would correctly trace out the flow of cerebrospinal fluid from formation to drainage? Let's look at how that would occur. You have this network of capillaries, these blood capillaries that line the roofs of the ventricle, referred to as the choroid plexus. Plexus just means a dense interconnected network of. Now, if you were to look really close at one of these capillaries, you would see here's your capillary, your blood vessel, kind of looping around. Capillaries are the point of exchange in the cardiovascular system. And when blood is pumped into these capillaries, there's a pressure behind it. That's essentially the job of the heart, is to create a blood pressure, right? It's to create a pressure gradient that moves blood through our blood vessels. When that blood, a little bit of it gets pushed across the capillary wall, it then hits what are called ependymal cells. Now, the extracellular fluid in the cardiovascular system is called blood plasma, but when that plasma gets pushed across the capillary wall, it undergoes a transformation, right? It's chemically a little bit different as this filtration process occurs. And once it gets through that entire filtration process, we're going to call it something different at the other end. So that blood plasma gets forced across the capillary walls. It comes into contact with these ependymal cells. They're connected via tight junctions, so not anything can get through. Those ependymal cells essentially filter that blood plasma, and when that blood plasma finally crosses those ependymal cells and enters into this volumetric space that we think of as being, in this case, the lateral ventricle, it's no longer called blood plasma, it's called cerebrospinal fluid, or CSF. So the formation of CSF happens at the choroid plexus, and it's facilitated to a large degree by the ependymal cells, which allow the blood plasma to be filtered prior to entering into these cavities. Now, we have the lateral ventricle. Cerebrospinal fluid flows from the lateral ventricle through this little hole called the interventricular foramen to the third ventricle. From the third ventricle, through this network right here, this hole or this tube, this canal called the cerebral aqueduct, to the fourth ventricle. From the fourth ventricle, that cerebrospinal fluid can either enter directly into the subarachnoid space, where it'll circulate around the brain and spinal cord, or it can enter into the central canal of the spinal cord, where it bathes the neurons of the spinal cord internally. It will eventually pop out the tip of the spinal cord, enter into the subarachnoid space, and that cerebrospinal fluid will circulate through the subarachnoid space. Now, as that cerebrospinal fluid circulates through the subarachnoid space, what it's doing is it's delivering things like nutrients and oxygen to neurons of the brain. It's rinsing metabolic waste and carbon dioxide. So it's giving the brain good stuff, the brain and spinal cord good stuff. It's taking away bad stuff. And as it begins to take away bad stuff, we have to get rid of that, and we have to filter all that bad stuff out at some point. What reabsorbs cerebrospinal fluid back into the cardiovascular system are these little structures right here, which are folds of the arachnoid mater, referred to as arachnoid villi or arachnoid granulations. So whereas the choroid plexus synthesizes cerebrospinal fluid, the arachnoid villi reabsorb cerebrospinal fluid. When they reabsorb cerebrospinal fluid, they reabsorb it into this big fat daddy vein with blood flowing through it called the superior sagittal sinus. Once that cerebrospinal fluid is reabsorbed back into the cardiovascular system, we no longer call it cerebrospinal fluid, we call it blood plasma. So we have from plasma through the choroid plexus and ependymal cells to cerebrospinal fluid, lateral ventricle, interventricular foramen to third ventricle, cerebral aqueduct to fourth ventricle, either out of the apertures in the fourth ventricle to the subarachnoid space directly or into the central canal. The common point of collection is the subarachnoid space. That cerebrospinal fluid circulates around the subarachnoid space where it's eventually reabsorbed by the arachnoid villi. Now, you have a question on a short science article that I've given you that was looking at hydrocephalus post-viral infection. And when you think about that hydrocephalus that develops post-viral infection, let's think about the words there. So hydro means water and cephalus means brain. What you're talking about is an excess of fluid on the brain, and I can't see the article. It's not directly in front of me. And then it talks about reovirus type 1 infection in mice. So they inoculated these mice with an infection. They gave them a viral pathogen, and they looked to see whether there were changes in the cerebrospinal fluid circulatory routes. And they did, in fact, see those changes. In fact, what ends up happening is that the arachnoid villi become scarred over. There's fibrous tissue formation over the arachnoid villi. Well, when you think about what would happen if there was fibrous tissue formation over the arachnoid villi would no longer be able to drain cerebrospinal fluid very effectively. If they're not draining cerebrospinal fluid very effectively, you're going to get the buildup of cerebrospinal fluid. The buildup of cerebrospinal fluid is going to put pressure on the brain and spinal cord, and that's not going to be a good thing. So think about what would happen with respect to the pressure of fluid flowing in that cerebrospinal fluid. We call it intracranial pressure. If there were problems with the arachnoid villi as opposed to if there were problems with what was going on in the choroid plexus. Now, when we think about cerebrospinal fluid formation, we have these blood vessel networks of the choroid plexus filtering through those ependymal cells, and you can kind of see the way that cerebrospinal fluid spreads out. So if you look at this GIF one more time, it starts in the lateral ventricle, and then third ventricle, cerebral aqueduct, fourth ventricle, out that aperture into the subarachnoid space or into the central canal, out the tip. You know what I mean? And you can see it kind of spreading along, and then you can see the reabsorption of that cerebrospinal fluid back into the superior sagittal sinus. So this is just a nice image kind of emphasizing how that cerebrospinal fluid flows. It's a dynamic process. Now, when we think about the three functions of cerebrospinal fluid that I want you to know, is it provides buoyancy. So the brain and spinal cord physically float in cerebrospinal fluid. They don't come into contact with either the vertebral cavity or the cranial cavity. They're floating on a thin layer of cerebrospinal fluid. So it provides buoyancy. Cerebrospinal fluid delivers nutrients and oxygen to the tissues that make up the brain and spinal cord, and it removes things like metabolic waste and carbon dioxide from the tissues. Now, at any given point in time, we have about a half a can of soda of cerebrospinal fluid. We don't have very much, but we turn it over constantly throughout the day, meaning that we're constantly draining and remaking cerebrospinal fluid. Where you get most of the turnover of cerebrospinal fluid is actually while you're sleeping. So you can think of one of the reasons that you don't feel good if you don't get a night's sleep is that cerebrospinal fluid remains relatively stagnant. You start building up things in that cerebrospinal fluid that interfere with the brain being able to function efficiently. Now, when you look at this sagittal section of this MRI, you'll actually see that the brain doesn't actually contact the cranial cavity. It's physically floating on that layer of cerebrospinal fluid. In addition to that, we can see the meninges really clear, especially the dura mater. So there's the dura mater, there's the subarachnoid space right there with the cerebrospinal fluid flowing. Just gives you an appreciation for the imaging when you kind of learn what you're looking at. Now, cerebrospinal fluid is used all the time diagnostically. We use cerebrospinal fluid pretty consistently from a diagnostic perspective. The way that we acquire cerebrospinal fluid is we have somebody usually lay down in the fetal position. We look for the splayed out spinous processes of usually, I think it's the L4 and L5 lumbar vertebrae. I may be wrong on that. And you insert a needle into that area in order to extract fluid. One of the things we care about when we're looking at cerebrospinal fluid is pressure. We call it intracranial pressure. And what happens is we have a device very similar to what you use on like a car, and if pressure is elevated, you'll see it go whoop, and you'll see the little probe on that device go up to indicate that there's a high fluid pressure in there. Another thing we care about is we just look at that cerebrospinal fluid and we look for turbidity, coloration. Xanthochromia, which is where you get cerebrospinal fluid that's a little bit yellow, can indicate something like a bleed in that cerebrospinal fluid pathway, and that's not good. Turbidity, right, just it being a little bit cloudy could indicate something like a infection, right? Now, cerebrospinal fluid, I've been told, actually smells kind of like banana. That's what a military medic told me, that he was trained, if somebody's leaking out of their ear or something and it's cerebrospinal fluid, he was trained to smell it and see whether it smelled like a banana, at least in the conversation that we had together, which I thought was very interesting. We can also look for proteins, we can look for sugar levels, we can look at different cell counts. Glucose, as a general rule of thumb, the concentration of glucose in the cerebrospinal fluid should be about 60% of the concentration of glucose in the blood plasma. And glucose is really important in the cerebrospinal fluid because neurons rely on glucose to carry out cellular respiration or to make energy necessary to do the things they do to keep us alive. So the procedure is called a lumbar puncture. You just extract it and you can get a real nasty headache afterwards. So if you're ever told to lay down after you get a lumbar puncture, take them seriously, because if you don't, your brain will contact the cranial cavity wall, it'll squeeze all those nerves in the meninges, and it'll give you what's called an LP headache. And let me tell you from personal experience, it is not a particularly pleasant headache to go through. So those are the support structures of the central nervous system. Now let's talk about terminology. White matter. If I asked you which of the following best describes the anatomical composition of white matter is made up of myelinated axons. So white matter is made up of myelinated axons. It is an umbrella term for wiring in the brain and the spinal cord and in the peripheral nervous system. They're wires. That's what myelinated axons are. They transmit information in the form of electrical signals called action potentials from point A to point B. Gray matter, on the other hand, is made up of dendrites, neuron cell bodies, and to a lesser degree unmyelinated axons, but I'm going to focus on the dendrites and the neuron cell bodies here. So whereas white matter is made up of myelinated axons, gray matter is made up of dendrites and neuron cell bodies. So white matter is like wiring that transmits information from point A to point B. Gray matter is kind of like processing center. That's where that information is processed or sense is made from that information. Integration takes place there, the processing of information. If I gave you a question similar to USB cable is to computer processor as blank is to blank, think about how you would answer that. What's white matter like and what's gray matter like in that analogy? So if I was to say, which of the following would be true of the anatomical composition of the substance indicated by the pointer? On the outside of our brain, the entirety of the external regions of the cerebrum, what we think of as being our higher order brain center, is made up of gray matter, and that gray matter is referred to collectively as being the cerebral cortex. Humans have a very thick cerebral cortex proportionate to their body size and very big brains as well. And remember that gray matter, right, matters. It makes a difference with respect to the functional properties of the nervous system that you're talking about. If gray matter is like processing center, the more gray matter you have, the more interesting capabilities that particular brain is going to be able to, the more interesting things that brain is going to be able to do because it just has more processing center. White matter, on the other hand, is myelinated axons. White matter, again, is an umbrella term for wires that transmit information from point A to point B in the nervous system. So when you think about gray matter in the central versus the peripheral nervous system, a bundle of gray matter, a discrete unit or bundle of gray matter that's responsible for mediating some specific set of activities in the central nervous system is referred to as a nuclei. So a bundle of gray matter that functions like a processing center in the central nervous system we refer to as being a nuclei. Not the nuclei of an atom, not the nuclei of a cell, but a control center that's made up of many thousands to millions of neurons that exists as a discrete or independent entity within the central nervous system. We refer to that as being a nuclei. So if you go up the brainstem here, you'll see the abducens nucleus, the motor nucleus of the trigeminal nerve. Those are all little chunks of gray matter that are serving a function. They're responsible for a specific set of functional tasks. Gray matter in the peripheral nervous system, little functional units of neuron cell bodies and dendrites in the peripheral nervous system, are referred to as ganglia. So we're going to see things like the dorsal root ganglion, the sympathetic chain ganglion. Just know that a ganglion is a chunk of gray matter in the peripheral nervous system that's responsible for carrying out a specific set of functions. So central nervous system, chunks of gray matter are called nuclei. Peripheral nervous system, chunks of nuclei or chunks of gray matter are called ganglia. Now, a tract is a bundle of myelinated axons within the central nervous system. So if we're talking about the transmission of information, the movement of information from point A to point B in the central nervous system, that information is transmitted along myelinated axons. And myelinated axons, bundles of myelinated axons in the central nervous system that are carrying similar groups of information, we refer to as tracts. Bundle of myelinated axons in the peripheral nervous system, on the other hand, so once those axons exit the central nervous system and they form part of nerves, we refer to as nerves. So a bundle of myelinated axons carrying information in the central nervous system is a tract, whereas a bundle of myelinated axons carrying information in the peripheral nervous system is called a nerve. And remember, the peripheral nervous system is made up of your spinal nerves, your cranial nerves, the associated ganglia, sensory receptors, and neuroeffector junctions. So what is the connection between tracts and nerves? Well, the neurons that make up tracts synapse or communicate with the neurons that make up nerves all the time. And in that way, the peripheral nervous system can communicate with the central nervous system, or the central nervous system, in this case, in this motor pathway, can communicate with the peripheral nervous system. Now, when we think about the functional organization of the spinal cord, so now we have some key terminology down. White matter, which is made up of myelinated axons. We have gray matter, which is made up of dendrites, neuron cell bodies, and unmyelinated axons. White matter is essentially wiring that transmits information from point A to point B in the central nervous system and peripheral nervous system. White matter of the central nervous system is referred to as tracts, right? Of the peripheral nervous system is referred to as nerves, so those are white matter structures predominantly. Gray matter, on the other hand, is made up of neuron cell bodies and dendrites, and this is where information processing occurs. Now, remember that the spinal cord is only about the thickness of your pinky finger. If we take a transverse section of the spinal cord and look at it under a microscope, that WM means white matter and that GM means gray matter. One of the first things I absolutely positively want you to do, no matter what, when you get an image of the spinal cord, especially a histological image, is I want you to distinguish the anterior portions of the spinal cord or the ventral portions of the spinal cord from the posterior or dorsal portions of the spinal cord. And you do that in a couple of different ways. My preferred approach is to look at a vertical groove that runs the entire length of the spinal cord called the ventral median fissure or anterior median fissure. Remember, ventral and anterior can largely be used interchangeably. And again, that's that vertical groove that moves runs the entire length of the spinal cord. So now we know where we're looking at the anterior portion of the spinal cord here and the posterior portion of the spinal cord here. When we think about the spinal cord's organization, one of the most important things to determine right off the bat is anterior versus posterior, because the functional organization of neurons in the spinal cord is critically dependent on those regions, that regional kind of separation. So we're looking at a cross-section of the spinal cord, or a transverse section of the spinal cord here. Let's do some spinal cord anatomy. Identify the structure indicated by the pointer. That would be the ventral median fissure. Now, when we look at the gray matter structures of the spinal cord, the gray matter structures we care about, here we have what's called the anterior gray horn. Here we have what's called the lateral gray horn. And everything posterior to this, these big guys back here, those are called the posterior gray horns. You can also refer to this as the ventral horn, lateral horn, and dorsal horn. It's up to you. You can draw that preference how you will. Now, the little chunk of gray matter that traverses from one hemisphere of the spinal cord to the next is called the gray commissure. Running through the gray commissure, we have the central canal. The central canal is where cerebrospinal fluid circulates. So we have the gray matter structures of the spinal cord, these horns that kind of have this butterfly-shaped appearance. Then we have the white matter structures of the spinal cord. Now for this introductory class, this white matter structure right here is called the anterior white column. Here is the lateral white column, and here is the posterior white column. Now, gray matter is made up of dendrites and neuron cell bodies. This is information processing area. White matter is made up of myelinated axons. This is where information is traveling up and down the spinal cord, either to the brain or away from the brain. The first focal point we're going to draw is to the gray matter structures of the spinal cord, and we're briefly going to talk about the functional characteristics of each of these gray matter structures. Not in any degree of depth, but just a brief overview to give you a better idea. So, what you're looking at here is a bundle of myelinated axons in the peripheral nervous system. A bundle of myelinated axons in the peripheral nervous system is called a nerve. Now you'll notice that there's visceral motor neurons and somatic motor neurons. You'll also notice that there are visceral sensory neurons and somatic sensory neurons. Another way of saying a visceral motor neuron is saying an autonomic motor neuron. And remember, the effectors of autonomic motor neurons are cardiac muscle, smooth muscle, and gland. That's what autonomic motor neurons control. The effector of a somatic motor neuron is skeletal muscle, meaning somatic motor neurons control skeletal muscle. So we see our motor neurons here, the axons of motor neurons, and then we see our sensory neurons here. So visceral or autonomic sensory neurons detect things like stretch, chemical composition, things that we don't process largely at a conscious level, but that our brain is constantly needing input about in order to regulate our organs. And somatic sensory neurons, which transmit information that is processed at a conscious level, meaning it works our way up to our brain, works its way up to our higher order brain structures, and it's constructed into this conscious experience of touch or temperature or pain or tickle or vibration. Now, spinal nerves are called mixed nerves because they have both motor neurons and sensory neurons running through them. All spinal nerves contain both motor and sensory neurons. Now, let's focus on a sensory pathway. So let's say that you're pricked by a needle. That triggers a free nerve ending in the integument. It's detected, right, by the free nerve ending. It triggers a graded potential, which leads to an action potential. That action potential travels along this somatic sensory neuron. Now remember that all sensory neurons from the previous lecture that carry somatic sensory and autonomic sensory information are unipolar neurons. So unipolar neurons only have one projection coming directly off their cell body, and that's this guy right here. Because those unipolar neurons need a place for their cell bodies to exist, where their cell bodies exist is actually within this structure called the dorsal root ganglion, which is part of the afferent pathway or the sensory pathway. It's called a ganglion because it's essentially a bundle of neuron cell bodies in the peripheral nervous system. Those are the cell bodies of unipolar neurons that are responsible for carrying sensory information. So if I said which of the following is true of the dorsal root ganglion, well, it's composed predominantly of the neuron cell bodies of unipolar neurons because sensory neurons are unipolar. So we have our dorsal root ganglion. That information continues to travel along the axon of those unipolar neurons until it enters the spinal cord, and both that autonomic, aka visceral sensory information, and that somatic sensory information enter into the posterior gray horn. So the job of the posterior gray horn of the spinal cord is to take in sensory information and ultimately to route it to wherever it's going to go. So sensory information comes into the posterior gray horn of the spinal cord. Motor information projects out of the lateral and anterior portions of the gray horns of the spinal cord. And that's kind of a nice circuit. Sensory in, motor out, sensory in, motor out. It prevents overlapping circuitry. It's a good design. It's a good electrical engineering design, biological electrical engineering. So, we have our posterior gray horn, which receives all sensory information. Our lateral gray horn, if I was to ask you which of the following is true of the lateral gray horn? Well, the lateral gray horn contains the cell bodies and dendrites of what are called autonomic, aka visceral motor neurons. When activated, these autonomic or visceral motor neurons fire an action potential that then travels along what's called the ventral root. So whereas the dorsal root carries sensory information, the ventral root carries motor information. Information traveling along these visceral / autonomic motor neurons will eventually make their way to either cardiac muscle, where it'll influence the rate of the heart, smooth muscle, where it will trigger either contraction or relaxation, and things like the GI tract, your blood vessels, etc., are dependent on that. Or glands, such as sweat glands, for example. Right? So the effector of autonomic motor neurons is cardiac muscle, smooth muscle, or glands in the GI tract, whichever glands you're talking about, glands in the adrenal gland, for example, whatever gland. And these are the motor outputs that you don't regulate at a conscious level. You can't decide that you're going to start producing sweat along the sweat glands in your antebrachium or in your forearm. You don't have conscious control. You don't exert conscious influence over that. The anterior gray horn contains the cell bodies of what are called somatic motor neurons. And somatic motor neurons are different than autonomic motor neurons in the fact that somatic motor neurons, if you remember, are regulated by higher order brain centers. So somatic motor neurons are under conscious control. Somatic motor neurons project out, so the cell bodies of somatic motor neurons would make up essentially the anterior gray horn, and then they project out and they travel along the ventral root. These somatic motor neurons extend all the way out until they ultimately, innervate skeletal muscle. So somatic motor neurons are responsible for regulating the activity, forming the medium that the central nervous system communicates with the skeletal muscles in your peripheral nervous system. So the posterior gray horn is predominantly responsible for sensory functions. The lateral gray horn houses autonomic motor neurons, which regulate autonomic motor functions, and the anterior gray horn houses somatic motor neurons, which regulates the activity of skeletal muscle. Now, when you think about spinal cord injury, we'll think about some pathologies of concern. So, pretend there was an infectious disease spread through contaminated water, and in fact there is, polio. So poliomyelitis is an infectious pathogen spread through contaminated water, and what it is it's a viral infection that strategically targets neurons in the anterior gray horn. And if neurons in the anterior gray horn are destroyed, think about what would happen. What do neurons in the anterior gray horn do? Well, they control and ultimately allow you to contract skeletal muscle. So if neurons in the anterior gray horn are destroyed, right? You're not going to be able to regulate the skeletal muscle in that particular region or in that segment of the spinal cord anymore. And that can produce what's called paralytic polio. Now, paralytic polio, before we had vaccinations, was a massive public health concern. You think COVID-19 was bad? Paralytic polio, my goodness. So, what you're looking at here are children who haven't received vaccines for polio and they're suffering the consequences of the paralytic effects of having that region of the spinal cord destroyed. The interesting thing is that the sensory pathways often remain intact, so they don't get that extra little benefit of not being able to feel those areas of their body because it largely or is restricted to the anterior gray horns. Now, the spinal cord model we're going to be using is this guy right here. So we have the anterior median fissure, anterior white column, lateral white column, posterior white column, anterior gray horn, lateral gray horn. The lateral gray horns contain autonomic motor neurons. Those autonomic motor neurons ultimately regulate things like cardiac muscle, smooth muscle, and glands. Now, if you have a lesion or some kind of damage to the lateral gray horns, which can be caused by a heavy metal toxicity or vitamin deficiencies, what that's going to manifest as is what's called autonomic dysregulation. So let's say that this pathway, this lateral gray horn pathway, plays a role in regulating the smooth muscle contractions in the stomach. If that pathway is no longer working properly, you're going to have things like stomach issues and you're going to have indigestion and be kind of burping things up and you're going to go, oh, I wonder what that is. Do an MRI, you might see the lesion is on the lateral gray horn. So they can be kind of hard conditions to pick out because you're not consciously aware of them. You don't consciously actually contract the muscles in your stomach to move stuff along or inhibit the muscles in your stomach. So autonomic dysfunction can present pretty weird because we don't really think about it, but it's so important. So you could start, sweating out of nowhere and not being able to sweat, whatever it is, cardiac muscle, smooth muscle, or glands. The posterior gray horn, when the posterior gray horn is compromised, it often results in sensory deficits. So, putting your hand on a hot stove and not noticing it because that sensory information isn't getting to where it needs to go. And it makes sense that that pathology would be like it is just due to the actual anatomical and functional arrangement of the spinal cord structures. Now, the white matter structures of the spinal cord, remember a bundle of myelinated axons in the central nervous system is called a tract, and in the peripheral nervous system is called a nerve. The neurons that make up tracts communicate with the neurons that make up nerves all the time, and in this way, the central nervous system can get information to and from the peripheral nervous system out to the tissues of our body. And remember that the central nervous system tells the tissues of our body what to do, right? The nervous system is kind of an overarching system of homeostasis because it's capable of influencing other systems. Now, a couple of tracts you need to know. So the way we name a tract is we always name tracts based on their point of origin and their point of termination. So the corticospinal tract has a point of origin in the cerebral cortex, which is a conscious center in your brain, and a point of termination in the spinal cord. Any tract that has a point of origin in the brain and a point of termination in the spinal cord is what's called a descending tract, meaning information is moving down from the brain to the spinal cord. And all descending tracts are carrying motor information because why would you carry sensory information away from the brain? So you always name a tract where it originates and where it terminates. And if it originates in the brain and terminates in the spinal cord, then you're looking at what's called a descending tract, and that descending tract is going to be carrying sensory information. The most important of the descending tracts you need to know are what are called the corticospinal tract. Now, pyramidal tracts ultimately regulate essentially the activity of skeletal muscle. Pyramidal tracts are called that because these neurons that you find in the motor cortex, this primary motor cortex that you're looking at here, are what are called pyramidal neurons. So we refer to these tracts that originate from pyramidal neurons as being pyramidal tracts. The most important of the pyramidal tracts to know is the corticospinal tract. So these upper motor neurons project down, cross over at the level of the medulla oblongata, right? Meaning the right hemisphere of the brain controls the left side of the body, left hemisphere of the brain controls the right side of the body. And the neurons that make up these upper motor neurons, the axons of which make up the corticospinal tract, communicate with the somatic motor neurons in the anterior gray horn. This synapse or communication between the upper motor neuron and somatic motor neuron is essentially an exchange of information between the corticospinal tract, which originated up in the brain, and one of your spinal nerves. This somatic motor neuron, the axon, then projects out, forms part of a spinal nerve, and extends all the way to the skeletal muscle that it controls, or the effector of which is. And it tells that skeletal muscle, hey, contract. So we have the corticospinal tract. We also have, when you think about these, they're also called pyramidal tracts because they cross over at the medulla oblongata and they form these medullary pyramids. Now, corticobulbar tract is just neurons, upper motor neurons that originate in the primary motor cortex, and they communicate with lower motor neurons or somatic motor neurons in the brainstem rather than the spinal cord. And this is how, for example, the muscles in the somatic motor control of the facial muscles is regulated. So that's pointing out the same thing. Now, sensory pathways. Sensory pathways are named much the same way that motor pathways are, except in a sensory pathway, you're beginning in the spinal cord and you're ending in the brain. So sensory pathways will always start out with a spinal structure and end with a brain structure. So in this case, the one that you need to know is what's called the spinothalamic tract. Begins in the spinal cord, terminates in the thalamus. The spinothalamic tract actually interacts with these neurons, and we talk more about sensory perception, but they're called first-order sensory neurons, and they're associated with free nerve endings. So these free nerve endings are embedded in your integument. They pick up temperature and pain. When they're stimulated, an electrical signal called an action potential is triggered. That action potential travels along that unipolar sensory neuron, there's your dorsal root ganglion right there, to the posterior gray horn. This first-order sensory neuron then synapses with a second-order sensory neuron, which crosses over in the spinal cord itself, and it forms part of what's called the second-order sensory neuron, the axon, forms part of what's called the spinothalamic tract. And I'm trying to find that on this, spinothalamic tract, because it originates in the spinal cord and it terminates in a region of the brain called the thalamus. Second-order sensory neurons then synapse with third-order sensory neurons in the thalamus. The thalamus is kind of like a routing center, and we'll talk about that later, which transmits that information to the functional region of the brain that's responsible for processing that information. In this case, the somatosensory cortex. Remember, somatic sensations are sensations that are perceived at a conscious level, so temperature and pain would definitely be a somatic sensation. This would be called, the spinothalamic tract would be considered an ascending tract because it's carrying information from the spinal cord to the brain, and all ascending pathways by definition are sensory. So thank you very much for your time. Appreciate it very much. Have a good one until I see you again.